welcome everyone. This is our first Horizon case series. So the purpose of this series is to identify complex cases that usually don't allow you to, markedly diagnose the patient using the genomic pipeline. That requires more kind of a deep dive into the phenotype and into the genes and look into the variants. So this case series wants to bring out those complexities and also want to show how we can untangle those complexities by using tools and algorithms so we can have a biologically meaningful insight on genetic diagnosis. So today we have our first guest for the series, Dr. Hosnera Akhtar. She is the Director of Genetics and Genomic Medicine Center, GGMC, at Neurogen Healthcare. She did her PhD in genetics from Dhaka University, and we are happy to have you. So take it away, Dr. Hosnera.
Thank you, Dr. Mohammed, for your warm introduction and inviting me to speak in such a wonderful session. I'm Hosnera Akhtar. Welcome everyone in today's webinar. In this session, I will present a complex rare disease case and also try to highlight the modern genomics and the automated tertiary platform, which are now transforming rapidly into the medical diagnosis and patient care. So before starting, I will now show you some brief overview of Neurogen Healthcare. The Neurogen Healthcare actually only one of the genetic testing laboratory in Bangladesh, which has modern genetic testing capability and providing targeted panel-based sequencing, exome sequencing, and genome sequencing services for the diagnosis of rare diseases, neurodevelopmental disorders and neurological disorders, metabolic disorders and myopathies, cancers, and other genetic diseases. In this center, as we most of the patients are rare disease patients, that's why we are providing our services through a circle of care team. We have various types of professionals from different professionals like doctors, geneticists, genetic counselor, clinical psychologist and therapist on various disciplines like from speech and language department, occupational, physiotherapist, behavioral modification therapist, and certain types of babies need special food diet chart. That's why we have a well-skilled nutritionist in our center. So Neurogen starts its journey from October 2016. Now it is almost 9 years, and throughout this journey, Neurogen actually did more than 1,500 exome sequencing tests, over 400 targeting sequencing tests, and 2 whole genome sequencing tests. So we have so many rare disease cases from neurodevelopmental disorders, neurological disorders, and neuromuscular disorders. We have explored so many variants that are unknown and also very rare in our population throughout this research, doing research. So this is our Genetics and Genomic Medicine Center, from where we are providing all types of genetic testing services. This is the interior part of our biological sample preparation room, where blood and tissue samples are processed. And this is the library preparation and sequencing room. And this is a glimpse of our work environment. Our experts are from mainly from genetics, biotechnology, and molecular biology background. So now I will show you how we are interpreting clinical variant. I'll try to show you the process. So world standard guideline is American College of Medical Genetics and Genomics guideline for the clinical variant interpretation. So ACMG actually assigned 28 criteria based on evidence type, like the frequency of the allele, which you will get it from population data, and some in situ computational tools from which you will give an idea of predictive value for prioritizing variant, functional data, segregation data, de novo data based on allelic data, other databases like if the variant is submitted in ClinVar by others clinical laboratory, and other data whether the variant is actually associated with patient's phenotype. Based on this type, the 28 SCMG criteria again can be categorized into two types: pathogenic criteria and benign criteria. So there is 16 pathogenic criteria and 12 benign criteria. These criteria are again combined to assign the variants into 5 tiers, like pathogenic, likely pathogenic. Pathogenic means the variants which have conclusive evidence for causing the diseases. Likely pathogenic means the variants not have enough conclusive evidence but has strong evidence in favor of pathogenicity. And the variants which does not meet the criteria of pathogenic or benign, those variants are assigned as variant of uncertain significance, and the variants further two category variants are benign and likely benign variants. So now I will show you the detail of a rare disease case who was referred by clinician in our center. She was a 11-month-old girl and came to our center with the phenotype of developmental delay and recurrent focal epilepsy. If we go back to his past medical history, we can see that the baby was a cesarean baby and her birth weight was 3.5 kg. She had no prenatal complications, but soon after delivery, she developed hypoxia and she needs around 3 days oxygen support. At the age of 7 days, she developed neonatal jaundice. And at the age of 1 month, her parents noticed that her body is shaking during sleeping. And they took consultation from pediatric neurologist, and doctors actually administered her some anti-epileptic drug. After that, around 7 to 8 days later, the baby was found to have recurrent focal seizures, which was actually the time duration was around 30 seconds to 40 seconds, but it was found weekly 2 times. And day by day, this frequency was getting increased, and the baby was losing her speech. She actually can say 2 to 3 meaningful words at the age of 9 or 10 months, but when she came to our center, she actually lost that. She lost her gripping power, and her all motor milestones were delayed. So when she came to our center, our pediatric neurologist, our genetic counselor, and other expert did a thorough evaluation of her phenotype and suggested that the baby, it looked like that she is a girl baby, and the regression has been started. Maybe it is MACP2 Rett syndrome that was previously diagnosed in other centers, but our clinician actually didn't find enough criteria to set the phenotype into Rett syndrome because the baby had no deceleration in her OFC, and she has no stereotyping hand movement. That's why our genetic counselor and clinician suggested her parents to do exome sequencing to identify the causal variants. So after exome sequencing, we actually got 2 missense variants into different genes, one in voltage-gated potassium channel gene, which is KCNH1, and another in glucose 6-phosphate dehydrogenase gene. So now I will show you how actually we assign this variant into as a primary pathogenic variant in the next couple of slides. When actually KCNH1 variant disruptive variants in this gene actually associated with two known rare neurodevelopmental syndrome. One is Temple-Barrett syndrome, and another Gemelman-Leber syndrome. So when we found this type of syndrome, at first we could not concise that the patient actually the variant is actually associated with her phenotype, but her other neurological phenotype like focal epilepsy, hypotonia, regression, motor delay, all this overlap with this syndrome. Then we look back into some paper and try to find out the detailed phenotype of the KCNH1 variant patients. So we got a paper, actually not a single paper, multiple paper, and found a detailed phenotype of the patients. If you look at the table, you can see that a study where 51 different types of epilepsy patients were taken, and they had 30 KCNH1 variants. So of the 30 KCNH1 variants, 5 patients actually had the same variant which was found in our patient, and you can see that the phenotype of the epilepsy spectrum was from generalized tonic-clonic seizure, focal epilepsy, febrile seizure, and even status epilepticus. And another thing is that out of 5 patients, 4 patients had Temple-Barrett syndrome, and 1 patient who had only focal seizures with intellectual disability and developmental delay. So then we tried to see actually which types of domains are involved in only focal seizures, and we got some idea that there are 2 transmembrane domains which is actually called hotspot variants, hotspot region for missense variants, and if the variants occur in these regions and the main phenotype is epileptic encephalopathy, but the TBS or JDLS syndrome, the variants actually scattered throughout the KCNH1 gene. So these types of criteria helps us to assign the ACMG criteria. So next, another variant, I'll talk about another variant, which is glucose 6-phosphate dehydrogenase, which occur glucose 6-phosphate dehydrogenase gene. Due to this variation, which is actually called G6PD Orisha variant, and it is the class 1 variant, and the patient actually glucose patient actually suffer with anemia because due to deficiency of glucose 6-phosphate dehydrogenase. So I did some literature actually what actually happened, and I have found the FDA approved database for pharmacogenomics drug where actually G6PD play role in the metabolism of G6PD. Sorry. So pathogenic variant in the glucose 6-phosphate dehydrogenase gene cause glucose 6-phosphate dehydrogenase deficiency that can lead to hemolytic anemia when exposed to oxidative stress. So this Orisha variant was previously found in one of our cohort, which is neurodevelopmental disorder cohort, and we found that same Orisha variant was present in the patients of autism, ADHD, seizure, behavioral abnormality, etc. Patients with glucose 6-phosphate dehydrogenase should avoid some known oxidative drugs like some drugs which are metabolized through oxidative pathways like anti-malarial drugs, sulfonamides, nitrofurantoin, and certain analgesics and other medications. Also, the patients should avoid some foods like broad beans and certain legumes, and which actually triggers the oxidation, and some infections also triggers the oxidation. That's why the patient should the patient's family should take care in this aspect. So which criteria actually set with KCNH1 variant? We actually, as our patient had focal epilepsy and others abnormality that match with the established patients' clinical phenotype, we assigned actually 6 criteria and combined 6 criteria and ultimately assigned the variant as pathogenic. Actually, this variant was previously established as a pathogenic variant, and this variant was not found in population database. We actually didn't get a chance to do the family analysis. Rather, we assumed that the variant is de novo at literature also support this. That's why we assigned it as a PM6. And there are multiple lines of computational evidence that support that the variant has a deleterious effect on the gene product. And patient's phenotype were highly specific for the gene, and the reputable source ClinVar has multiple entry for this variant as pathogenic and likely pathogenic. So another variant that is like pharmacogenomic findings, glucose 6-phosphate dehydrogenase, it was assigned as likely pathogenic, and the SCMG criteria PS1, PP3, PP5 align with this criteria. Now, as you know that in whole exome sequencing, there are on an average 80,000 to 100K, 80,000 to 100K variants. So streamlining this variant and prioritizing the variant is very troublesome. So our lab is actually a busy lab. We have to provide around 60 to 80 reports per month. That's why we actually needed a automated platform for doing the tertiary analysis. That's why we do the partnership with GenomeArc. Now I will show that the case that I showed you actually the analysis, full tertiary analysis was done in the GenomeArc platform. How I did the analysis, I will now try to show you the overview. So what will you get actually in GenomeArc? At the very beginning, the phenogene module of the Horizon, what actually it's actually the unstructured phenotype into the human phenotype ontology term, HPO term. So from this, another thing it does, it actually faced the possible disease association, possible disease that match with the scattered phenotype, HPO terms, and it also provide another a set of genes associated with the patient's phenotype. So around 50 to 60 genes has been are found from phenogene module, and we initially do the analysis using this. Next, another thing that we need to set the SCMG criteria. So there is a workflow in the Horizon where we can set the various criteria by providing our SCMG computational value like PP3, and whether the frequency is aligned to PM2. You can set the Horizon has some integrated curated databases, so proprietary databases, which give you an idea on PS1 and PS4. So next, I will show you how we can upload the genomic file in Horizon. Actually, we need to, for tertiary analysis, we need to upload the VCF file in the genetic data upload section. And it is really interesting that the upload of VCF file takes only 3 to 5 minutes. Before using GenomeArc, we did the annotation of the VCF, and it took around 12 hours to 1 day, because it actually depends on server capacity of your center. When we started partnership with GenomeArc, we started the analysis in Horizon platform, and we really surprised that the VCF upload takes only 3 to 5 minutes. And as soon as the upload, you will get a summary report from the genome summary tab. and you don't need to do extra time to find out the pharmacogenomic, the variant which is related with pharmacogenomics, and is there any variants that need to do carrier screening, and is there any secondary findings where ACMG, you know that where ACMG 84 genes are has actionable then. So, and this summary will give you an overview on the genome. After that, in variant analytics tab, there is you can set initially set the standard workflow. Under standard workflow and clinical variant tab, you can analyze the data, and you will get around 30 to 40 variants, which is clinically relevant with the patient's phenotype. So what actually we get before when we did the analysis through traditional bioinformatics process, we actually have to set several types of filter criteria in the Excel file, and it took too much time to analyze the tertiary analysis. And initially we get around 500 to 600 variants, which need to do further analysis for to assign as with patient phenotype. So what actually I have found around 29 variants in the clinical variant tab, and at the very the first row you can see the G6PD, which has a pharmacogenomics role, and here is a tab where you can see that there is rare disease sections where the disease you can get idea the disease. And in my previous slide, I have actually told that the variant can be present in some somatic cell, which is sporadic, and that information we will get from this pan cancer tab. And there is gene insights, another tab, and at the later side you will get the ClinVar entry and population frequency tab. So next, which is our primary findings, KCNH1 variant. You can see that GenomeArc actually Horizon can first detect this variant as de novo from the tab where it has a proprietary databases where so many variants are curated from literatures. So actually, in my previous slide, I showed you a paper. That paper actually is this paper. Initially, when we did the traditional bioinformatic process, we actually searched the phenotype and gene in the Google and Google Scholar or PubMed, and they are actually spent so many times for interpreting so many variants. So actually, you will get the target paper here, PS, which actually ICMG criteria PS4, and Horizon Cancer Database is also there. So actually, this analysis only took for the full analysis from phenotype to genotype and reporting. It took only less than 30 minutes. So it is really amazing to do analysis in Horizon. So lastly, I already told my previous slides that when we did the analysis manually using previous bioinformatics flow, it took around 6 to 13 hours, even some days to complete the analysis. But when we started the analysis with Horizon, it reduces the time from few days to only few hours. It actually reduced, and nowadays we can feel that it actually reduced our times fourfold reduced there. So variant interpretation takes actually too much troublesome for the variant interpreter. So if as our lab is so much busy for providing critical patients report, and timely report delivery is very essential for clinical decision making. So automated system can actually help us to provide the report for a highly scalable, high throughput laboratory. And we are really happy to have Horizon in our analysis platform. So after that, we also generate the report from Horizon. You can see that the everything is if you make a template in the Horizon, you just need to put your information here from the information tab, and the report is automatically generated, which actually previously need only report preparation after accumulating all the information from various databases. Report preparation took around 3 to 4 hours. But in Horizon, when you are doing the analysis, you just click this. Here are some tabs, and add to report. All the information will be found in the report, and you can easily print out the report. That's all. Thanks all the audience for your patience, and thank you.
Thank you, Hosnera, for such a deep dive into this particular patient who had a manifestation of, spectrum of phenotypes, and you kind of identified two different variants. It's kind of shows how complex genetics can be along with phenotype. So I'm going to open up for discussion to our colleagues. Please feel free to ask questions if you have any. Yeah.
I just have one question.
Yes.
First of all, that was really great and really interesting to follow. I just wanted to ask, I just saw in the pharmacogenomics list there that there was some variant links to specific, so see drug links there. And I just wondered regarding how influential they are in your reporting, and then on maybe later patient care treatment plans. Like, how do you use these? That would be great to know.
Very good question. Thank you. Actually, the patient not only have this variant, she also has another pathogenic variant in KCNH1 variant. So actually, patient to patient genetic makeup different, and patient phenotype, what will be the progression of the disease not only depend on your primary findings, it also depend on your secondary findings and other pharmacogenomic findings. So there are certain drugs which are metabolized in your system. So if there is any mutation in the metabolizing enzyme system, so your drug will not be metabolized properly. Now, what actually G6PD do? G6PD deficient cell actually produce huge byproduct, oxidative product, and it will damage your red blood cell, and you will be in anemic all the time. Maximum time you will be anemic if you are not cautious. Take caution in your food items, in your infections, and like there are in the FDA actually approved for 41 drugs referred which metabolized via oxidative pathway. So as the baby has the G6PD deficiency, she is already vulnerable for red blood cell destruction and anemia. So if these drugs are actually given to the baby, the baby will fall in huge oxidative stress, and her phenotype will be actually very aggravative, and its disease will be progress rapidly. So actually in this way, if the pharmacogenomic section help the patient in disease management actually, and the clinical decision making.
That's great, and it's immediate as well. So I suppose it means that you don't have to go and do further research. Just the papers are available. That's really interesting. Thanks for that.
Thank you.
I have a question.
Yep.
So thank you so much, Dr. Hosniara. It was an amazing case, and how you found not just the primary but also pharmacogenomics. My question sort of extends on your answer to Maurice's question. So KCNH1 gene seems to be contributing directly to the primary phenotype. But then regarding the G6PD mutation, do you think it might contribute as well into the phenotype overall by adding more complexities? Not just, not just its relationship with the primary phenotype, but rather the phenotype of the patient?
Okay, thank you. Actually, glucose 6-phosphate dehydrogenase, what actually do? This gene actually represent in your red blood cell, and through pentose phosphate pathway, it actually did help in balancing the oxidative reactions to NADPH by producing NADPH. So G6PD deficient cell will produce oxidative byproduct, and it is already known by several studies that redox oxidative byproducts are associated with neuroinflammation, and neuroinflammation ultimately associated with autism, behavior abnormality, and cognitive disorder. In our last recent studies that was actually done in 2024, and the cohort size was 576 neurodevelopmental disorder patients, and in this cohort we have found G6PD Urisha variant in 1.8% patients. So these patients had, or some have, male patients had mainly autism and behavioral abnormality. We have found 2 girls who have also seizure and behavioral abnormality and other phenotypes, cerebral palsy. So actually, if a long time a baby get into the oxidative triggers, so here the baby will be anemic, and there will be oxidative products that may actually lead to neuroinflammation, and it may be a modifier of his phenotype. From our study actually support that, from our study as well as other studies.
Thank you so much.
Okay, I'm going to ask you a bit of a technical question on the analysis. So you said the platform Horizon, GenomeArc Horizon reduces 4-fold of your time. I'm just wondering, for a busy lab like yours, you initially said you have 70 or 80 samples per month to, that you have to spin out the reports. How important this time advantage for your lab that Horizon is providing?
Thank you, doctor. Thank you. Actually, in my previous slides, I tried to show you that we have to set the ACMG criteria into patients' phenotype and genotype, and we used lot of databases manually, and this took actually a lot of time to interpret the variant and match the variant with patient phenotype. But Horizon, which actually did that, it has lot of information on lots of automation, and it have so many databases that no need the variant interpreter to do separately in Google or PubMed or Google Scholar. I have shown you that there is a tab where all literature information, curated databases are there. There are CleanVar, there are OMIM database, there are so many computational tools that will help you to interpret whether the variant has a damaging effect on the gene, whether the variant is associated with a similar phenotype in other cohort, whether the variant is present in lots of cancer tissue, or whether the variant is ACMG guided secondary finding variant or pharmacogenomic. All things are in a single tab. All things are single window. You can take the decision, just click the variant and to take the decision at a time. In our center, we are doing with critical patients. We need to provide the report as soon as possible, actually within 3 weeks from sample collection to providing the report within 3 weeks. And there a long journey or a long procedure of laboratory works, primary analysis, secondary analysis, and then we do the tertiary analysis, where actually we have we need only a few hours, 1 to 3 hours, to analyze the variant and do the report. And we provide the report fast to the clinician, and it helps the clinician to take the clinical decision making and start their treatment plan.
It's excellent.
Yeah.
I think I'm not sure if you guys have any additional question.
No, that was very interesting.
Yeah, I think this is a very good start of the series. We had an amazing case to discuss, and the fascinating part is that it has a pharmacogenetic variant as well. So we went above and beyond just genomics, touch based on the patient kind of management a little bit, and it shows a multifaceted advantage of using automation and platform like GenomeArc Horizon. So I'm very glad that we hosted you, Hosnera, and it was a fantastic talk. And yeah, so I think we will be back soon again with few more interesting cases like this, and not just going to limit on rare disease. We'll also be talking a lot of cancer cases that are complex and hard to pin down the genetic determinants. But yeah, this series will go on. It's a very fascinating resource for clinicians and for scientists, not just to understand the biology of the cases, also understand that application of technology, how this helps patient care. All right, thanks guys, thank you so much.
Okay, thank you. Take care.
Thank you everyone.